Hello everyone. Welcome to the Whole Genome Bisulfite Sequencing WGBS Data Analysis Learning Module. This is an introductory video of this learning module, why we designed it, and how to use it. This learning module was developed by the bioinformatics core of the Department of Quantitative Health Science in the University of Hawaii at Manoa. This WGBS learning module was among a diverse of learning modules that were developed under the support and effort of NIH NIGMS to convert biomedical research training materials into cloud-based learning modules. These learning modules covers various topics such as image analysis, metagenomics, epigenetics, and many others. These cloud-based learning modules contains course materials, useful tips, quizzes, and other resources which were stored and delivered through the Google Cloud platform. This allows students to access the materials from anywhere with an internet connection, with the benefits of greater accessibility, cost savings, and flexibility in the learning experience. The reason we chose the WGBS as the topic of this learning module is that DNA methylation is the most researched epigenetic mark involved in various processes in the human cell, including the development of many diseases. WGBS is a well-established protocol to determine the DNA methylation status of single cytosines on a genome-wide scale by treating the DNA with sodium bisulfite using high-throughput sequencing technologies. The data analysis of such sequencing data can help researchers to gain valuable insights into gene regulation and identify potential biomarkers in many disease processes. So what is DNA methylation? DNA methylation refers to the addition of a methyl group to the fifth carbon on the cytosine ring, giving rise to 5-methylcytosine. The whole process is mediated by DNA methyltransferases, DNMTs. DNA methylation is a critical biomedical process that happens millions of times in every single cell of the human body. In mammals, DNA methylation is essential for normal development and responsible for a vast range of biological functions, such as genomic imprinting, transposable element silencing, stem cell differentiation, embryonic development, X chromosome inactivation, and alternation in DNA methylation can also lead to aging and diseases such as cancer, atherosclerosis, nervous disorders, and cardiovascular diseases. To measure the DNA methylation level, bisulfite treatment was used to treat the genomic DNA before routine sequencing, and this treatment will convert cytosine residues to uracils, which leaves 5-methylcytosine residues unaffected. To be more specific, upon bisulfite treatment, unmethylated cytosines are deaminated to uracils, which upon sequencing are converted to thymidine. Simultaneously, methylated cytosines resist deamination and are still read as cytosines. In this way, the location of the methylated cytosines can be determined by comparing treated and untreated sequences. Here is an example of a WGBS study. The use of 2 kinase inhibitors enables derivation of mouse embryonic stem cells in the pluripotent ground state. Using whole genome bisulfite sequencing, as in our tutorial 1, we can show that the male embryonic stem cells treated with 2 kinase inhibitors are globally hypomethylated compared to the conventional embryonic stem cells maintained in serum. This data can provide genome-wide analysis of the dynamics of the deposition and removal of DNA methylation during the mouse early developmental processes. In this WGBS learning module, we will start from the FASTQ sequencing files and follow the workflows in the learning module to convert these sequences read into DNA methylation levels. And if more samples provided, we can further detect differentially methylated regions and genes of interest. The learning module will be provided in the format of Jupyter notebooks, and they can be downloaded from the NIH GitHub repository. Users will also need a Google account to access the Google Cloud Platform and run these notebooks on the GCP. The learning module mainly consists of 6 files. The first is the README file. The README file provides instructions such as how to download the module, how to set up the Google Cloud Platform, and all the other prerequisites. The next file is the Introduction PDF file. It offers the basic scientific background of DNA methylation, WGBS, and the steps to analyze WGBS data. This is more theoretical comparing to the real data analysis modules following. The first data analysis submodule is Tutorial 1. We also refer to it as Notebook 1, Lesson 1, Submodule 1. This notebook will use a small dataset and analyze it using Bismarck workflow step by step. The second submodule, Notebook 2, is to use the output from Notebook 1 and detect the differential methylated regions using a tool called Methylation. The third submodule is to use the same small dataset as used in Notebook 1 and analyze it using a Nextflow pipeline, Methylation. And the last submodule is Notebook 4. It uses a real dataset, much larger in size, to show how to run Methylation using Google Life Science API. Here is a more detailed architecture design. First, we have a scientific background introduction module. This is a document that can be viewed without a Google account. All the other submodules will need the GCP to view and run. The submodule 1 to 3 will be running using Vertex AI Notebook, while submodule 4 will utilize the Google Life Science API for larger dataset, which normally cannot be handled by the Vertex AI. For the first submodule Notebook 1, it will take a small example dataset as input, and after a step-by-step Bismarck workflow, the DNA methylation levels will be generated as output of the module. The second submodule Notebook 2 will take the output from Notebook 1 and process it using Methylation to detect DMRs. The output will be in the format of bed graph files and stored in the cloud storage. For Notebook 3, the same dataset that used in Notebook 1 will be used, but it will be analyzed through a different pipeline called Methylation, which is designed using Nextflow. The output should be in the same or very similar as results from the Notebook 1. As for the last submodule, a real-world WGBS dataset is used to give the students an example how to run large dataset on GCP. The jobs will send to the Google Life Science API, which provides an option to process large data at scale. The output files will be stored in a customized cloud storage bucket and can be used for other downstream analysis, such as the one from Notebook 2. To use the learning module, download the files we mentioned earlier and open them in the GCP Vertex AI notebook. All the information will be displayed in the Jupyter Notebook cells. There are 2 types of cells, Markdown cells and code cells. Follow the instructions in the Markdown cells and try to understand the information provided here. Then, run the code cell using the Run button on the top of the notebook, or by clicking Shift and Enter at the same time. The code in the code cell will run, and the output will be displayed below the code cell. If any error occurs, the error messages will also appear in the output. You can edit and add more code cells to test and explore the content from the learning modules. There will be quizzes and flashcards in some of the modules to help the students better understand and remember the contents. Well, that is all for the brief introduction of the WGBS learning module. Hope you can enjoy using it and learning the WGBS data analysis all by yourself. Thank you.